From the DZNE, the German Center for Neurodegenerative Diseases, where we put together a panel of our projects. I'm leading this session together with Marc Beyer. My name is Anna Aschenbrenner. We are both group leaders at the Systems Medicine Department at the DZNE in Bonn. And together with our colleague Omid Shirvani, who is an MD and clinician scientist in our department, training neurologist, we will showcase some of our projects here. Omid will start with introducing our NEUROCOV study. And please.
My name is Omid Shirvani. I'm a clinician scientist from the University Clinic of Bonn, also working at the DZNE. And I'm very happy to present today some preliminary data on an observational study that we are currently conducting in the context of our international NEUROCOV project. So NEUROCOV is an international project that is conducted across several countries in Europe plus Israel. And there are many scientists involved, especially from our center, the DZNE, that is currently doing the lead by Professor Schulze. And I'm talking here on behalf of the clinicians and neuropsychologists that are currently conducting the prospective and controlled observational study on patients with neurological kind of post-COVID at the Department of Vascular Neurology in Bonn and at the Department of Neurology and Experimental Neurology at the Charité. What are our inclusion and exclusion criteria? So we are currently including patients from 18 years with long-lasting symptoms after the infection, including subjective cognitive decline, and all of them have to present with an initial confirmation of the infection itself. For controls, it's necessary that they have no long-lasting symptoms after the infection. We have a few exclusion criteria. They shouldn't have a COVID-19 disease within the last 4 weeks, shouldn't be pregnant or breastfeeding. They should be sufficient in German because the neuropsychological testing that we are doing is in German. No one should participate in any other clinical drug studies. And for patients, this is very important for us, they shouldn't have any differential diagnosis that is currently explaining the symptoms. Our study runs for 12 months for each participant with 3 visits. The first visit at baseline, so that's the visit where we include our patients into our studies and also the healthy controls. We basically record data on the course of the infection. We record demographic and other clinical data of our patients, such as medication and past medical history. We do a neurological examination. We do a very comprehensive test battery of neuropsychological testings. This usually lasts 2 to 3 hours. And we provide our patients with self-report questionnaires asking for instance, general health aspects, symptoms of depression, and many other important symptoms in this cohort. We are also collecting blood samples, and if consent is given, we also do a spinal tap test. After 3 months, we see our participants again, collect again symptoms, medication, and blood samples. And after 12 months, we do the same, plus again a neuropsychological testing, and we provide them again with the self-report questionnaires. So this is preliminary data that I'm showing now. We have by now included 200 participants at Bonn in Berlin. And what you see is that in average, our patients are around 45 years old, and this is very much in keeping with the current literature on patients with long-lasting symptoms, neurological long-lasting symptoms after COVID. What you see is that the majority of our patients is female, and interestingly, there is a significant difference of premorbid conditions between both groups. So as you see, 36% or 37% of our patients had psychiatric conditions in the past and chronic pulmonary diseases at the day of inclusion. I'm going to show you now some symptoms that they have had during the infection and that they had at the day of admission to our hospital. During infection, patients report of having more symptoms, but this could be biased because they are still affected by those symptoms, and that's why they may memorize those symptoms better than controls do. Looking at the current symptoms, you see that in average our patients are reporting of having 7 symptoms, and they are mainly pain, such as headache, muscle ache, joint pain. They are also reporting of sensory disturbances. Another word for it is paresthesia. It's a tingling feeling on the skin. And they also have, in more than 90% of cases, fatigue, and this is not surprising, cognitive deficits. This is one of the inclusion criteria, and what they apparently demonstrate the most often is subjective concentration deficits, memory deficits, and speech difficulties. Now let's have a look on the neuropsychological test results. 1/3 of our patients, they demonstrate an impairment of cognition. And how is this defined in our study? We have different neuropsychological tests, and as soon as there are 2 tests with a 1.5 standard deviation below the mean of the corresponding same cognitive domain, we define this domain to be impaired. The majority of our patients is having a single domain impairment, and this especially affects attention. But there are also deficits in executive function, memory, and language. Comparing now those results with our controls, we see that there are 3 tests positive yet. One test is the test for alertness, in other words, reaction time. The other one is sustained attention, in other words, concentration, and both tests correspond to the cognitive domain of attention. And then the third one is the delayed verbal recall, in other words, memory. And this is also in keeping with the current literature on those patients, because they apparently are suffering a lot in those 2 domains. So attention and memory difficulties are a highly prevalent symptom in post-COVID patients. We have 2 tests to assess attention in patients, at least in our study. One is the TMT, it's a paper-pencil test, it's a tray-making test, and the other one is the test battery for attention assessment, and that is computerized. The first one is a very simple one, so basically you ask your patient to link letters or numbers in chronological order, and you stop the time. The second one is a bit more complex. Your patient, they see certain patterns or hear a certain sound, and then they have to push the mouse pad. And you basically measure the number of omissions or the reaction time. And we wanted to know which test is more appropriate for our patients, and checked how they classify our participants. And even though there's no significant difference, we see a tendency for the TAP test, and we think it's not significant yet due to the sample size of our group. We continue to analyze the association of fatigue, which is a very prominent symptom in patients, with the TAP test and depression. The TAP subtest of sustained attention, so you must know that the TAP test is divided into different subtests, and one of them is the sustained attention test. The sustained attention test was very interesting for us because we did not only see that patients are presenting with a higher rate of omissions and reaction time, but also the increase of omissions and reaction time over a period of 15 minutes was higher in those patients than in controls. And this was indicating that fatigue plays an important role here. Looking at correlation analysis, we see that the fatigue severity scale, we heard today results on the Shelder fatigue scale. There's also the fatigue severity scale, and it basically assesses the severity of pathological tiredness in patients. This was moderately and significantly correlated to the subtest of sustained attention. And in conclusion, we would say that the TAP test is more appropriate for patients due to different advantages. First of all, it's capturing fatigue. It might have a higher discriminatory power. It's inclusive for analphabetic individuals, And the best advantage is here, especially for our very severely affected patients that are bedridden, it can be done from home. Another very interesting finding from this correlation analysis was that the BDI-2, it's a self-report questionnaire that is assessing the burden of depressive symptoms, was highly and significantly correlated to the fatigue severity scale. Looking into the BDI-2, you see that more than 50% of our patients are suffering from at least mild symptoms of depression. 10% are even severely affected by symptoms of depression, including suicide thoughts. We then went to our colleagues from the Department of Psychiatry and asked to have a look at results of neuropsychological testings in patients with major depression. And this was basically the distribution of cognitive deficits in patients with major depression. So as you can see, they are mainly affected in attention, memory, and executive function. They unfortunately didn't measure the other cognitive domains. And then we wanted to compare the distribution of pathological findings in our patients in regards to those domains with patients with post-COVID, depression. So in other words, this is the distribution of depressed patients. And now I'm going to show you the distribution in post-COVID patients. And what you see, it's almost identical. This is in general leading us to a very important question. And as you know, I'm affiliated to the DZNE, and the DZNE is focused on neurodegenerative diseases. What we know is that depression precedes neurodegenerative diseases decades before the diagnosis is given. This is well known in Parkinson's patients, and this is also known in patients with Alzheimer's disease. Those are the 14 modifiable risk factors for Alzheimer's disease, and one of them is depression, and it usually occurs during the midlife. And there's also on the other side this debate whether psychiatric diseases or psychosomatic diseases may be caused by antigen exposure, of course, beside of many other kind of etiologies. And I want to leave you basically with two open questions. One is, how is antigen exposure, or in other words, infections, linked to mental disorders, cognitive decline, which is, by the way, well known in patients with depression. We call it pseudodementia. And decades later, neurodegenerative diseases. And could it be that patients with very similar phenotypes, such as post-Lyme disease, ME/CSF, post-COVID, and many others, depression, could it be that some of them are also suffering from a post-viral disease? So, in summary, the majority of our patients is female, around 45 years old and well educated. They primarily suffer from pain, sleep and sensory disturbances, fatigue, depression and cognitive decline, mainly inattention and memory. The TAP test serves as an efficient diagnostic tool that could be even done from home. The open questions that we want to basically answer is, could it be a potential risk factor for neurodegenerative diseases? And the complexity of symptoms, and this is really important because they are not only suffering from cognitive decline but also from psychiatric symptoms, pain, and many other things, needs an interdisciplinary and multimodal therapeutic approach. So thank you very much for your attention, and I'm happy to take your questions.
Yeah, Omid, thanks so much. Are there questions?
Yeah, bitte.
So I have one question from online,
Yeah.
which is basically there's a question asking about what tests you're using to evaluate depression, and if you're using the PHQ-9 test, and if so, that could have a bias. So that could you comment on that?
So we are currently using the BDI-2. Yeah.
So
it's not the other one. Yeah.
Okay.
Just out of curiosity, many in the study you showed that it's females and above 45, and of course this could be postmenopausal women as well.
Yeah.
Do you have a comparison cohort with postmenopausal women with similar symptoms maybe, and then see whether they share same signatures, or is this even overlapping?
That's a very important question. Thank you very much. And it was just recently also raised up at the DZNE. We have not particularly checked for signatures now as a comparison in women with postmenopausal symptoms. But of course, we have to keep this in mind, and we are currently discussing strategies. And possibly some of the symptoms could be explained by postmenopausal different changes in the body of women. However, as we see, there are also younger women affected, plus men. This must be something that is not primarily driven by postmenopausal changes. But of course, we have to take this into consideration, and it's very important, yeah.
Could be a subgroup.
Could be, yes, you're right, yeah.
Hi, thank you very much. I was wondering how you stratify this huge group of patients you're looking at. So do you actually look at post-exertional malaise or at ME/CFS patients? And I think if I didn't understand it wrong, you said that the cognitive tests tend to take up to 4 hours?
Yes, 2 to 3 hours, and we know. Yeah.
So are you aware that you might be missing many patients that are not able to perform those
Yes,
tests?
definitely. That's one of the limitations of our observational study. We are currently excluding those who are not able to get to an outpatient clinic for those kind of very exhausting testings. You're absolutely right. We have some strategies to help here. So what we offer is we have several breaks, or to do it within several days. But of course, this is a neglected subgroup of patients that we are not currently including into our study. Yes.
I think it would be very important to also clearly mention this when presenting your results and making it clear. Thank you.
By the way, this is why we are emphasizing the necessity of tests that can also be done remotely. So something like the TAP test could be done remotely from home and wouldn't need any more to get to an outpatient clinic.
Okay. Maybe Omid,
Yeah.
you can try to see what you can make of this question.
Which one?
The last one.
What is depression if not neuroinflammation in a subset, especially in long COVID? Why do we prescribe CBT instead of trying to prevent development of, e.g., cognitive decline after antigen exposure? I think you must know this is a huge question. So, I mean, in general, psychosomatic diseases need to be addressed by different kind of strategies, such as psychotherapy, and in some cases also medication. And what we are currently asking is the question if there could be a link to an antigen exposure, and what would be the appropriate therapy here. Is it psychotherapy? Is it a drug? And I think the most important, or one of the very important aspects that we are going to see in the next future is the biomarker profile of our patients. And this might help us also to stratify who could benefit the most from different strategy for therapy.
All right, for the sake of time, we will continue now with the next talk. Thank you, Omid, once again. Marc Beyer will now show a few further insights into our NEUROCOV study, which will follow on the samples, as well as mention some of our other projects in the DZNE.
Yeah, Anna, thanks so much for the kind introduction, and thanks a lot for having the opportunity today here to present, on the one hand, some of the molecular aspects that we try to address in the study Omid has just introduced, and at the same time also highlight what basically DZNE has been trying to do over the last few years concerning long COVID research. And we've been involved in multiple studies now over the last few years,
which also Anna, to some degree, afterwards will show. So we've been working in a large network across different study centers in Germany to collect blood transcriptomes and to identify blood-based biomarker for post-COVID. We've been also looking both in a DFG consortium as well as in a BMBF-funded consortium on the single-cell level, on deviations in the cellular composition and the molecular phenotypes within in blood cell types in post-COVID patients. We are also part of the biomarker platform of the National Clinical Study Group that has been introduced, that's led by Carmen Scheibenbogen. And lastly, we are leading this European consortium, this NEUROCOV consortium, where Omid has introduced, in principle, the German arm of the clinical study we're running there. And for the National Clinical Study Group that's led by Carmen Scheibenbogen, there are, in principle, 5 clinical trials that are using currently available therapeutic options, and in a double-blinded fashion, or at least for 4 of them, we're assessing, in principle, whether these treatments would benefit patients. And the primary readout there, of course, is physical activity, so whether that improves, but we are complementing this on the other side with biomarkers. And these studies, on the one hand, the first 2 here at the top are basically using immunoabsorption. I mean, we heard about that already today for ME/CFS patients. And the third one is using prednisolone as an immune modulator to also establish whether treatment with prednisolone would also benefit individual patients with cognitive deficits in post-COVID-19 syndrome. And in the very long study, vericiguat is given to individual patients, and the last one is in principle using hyperbaric oxygen treatment. And on the clinical side, I'm currently not yet able to comment on these, as the studies are just being completed or in the process of being completed during this year. So this has taken quite some time, that really these studies are now at their final stage. And in the biomarker platform, which is headed by Birgit Sawicki from Berlin, we will be looking now in peripheral blood monocytes that are going to, or are being collected as we speak, before treatment and then after basically completion of the treatment, and we'll be looking at cell composition changes using flow cytometry and CyTOF. That's being done also by Birgit. That's basically mainly being done by Birgit's group, and we will be using single-cell RNA-seq in combination with antibody stainings to identify gene signatures that are potentially indicative, on the one hand, of individuals that benefit from the therapies, or also that we could identify patient subcohorts that would be suitable for specific treatment arms in this study, or also develop biomarkers which will predict treatment responses. And this work, I hope, we will finish until end of the year, early next year, so that we then can also report on the outcome from the findings of this study. But for the moment being, this is just a look into the future that we hope to see here, really for these therapeutic, potentially therapeutic options that we want to see here also on the molecular level. What changes, and really is this potentially indicative of patient subgroups and treatment options. And for the NEUROCOV consortium or the study we're running there that Omid introduced, this is in principle a European study which is led by us, the DZNE, which is composed of 10 international sites. And the idea here is that we want to investigate the neurological and neuropsychological complications due to COVID-19.
and want to understand on the molecular level what is actually driving this. And the first main aim of this is to characterize the cognitive and neurodegenerative complications resulting from post-COVID-19. So that was what basically Omid has introduced, and the German study in Bonn and Berlin is complemented on the other side with a study in Italy where we're recruiting the same number of patients and are doing exactly the same or almost the same clinical as well as molecular tests. We then want to also define the cellular molecular mechanisms underlying NeuroCOV disease trajectories. So from our specialty, we will be doing this on the single cell omics level, looking at PBMCs, so blood-based assessments of cellular composition and molecular changes. And this is complemented on the other side by the colleagues from Italy, which are generating iPSC cells from these individuals, are then generating brain organoids and will be stimulating these brain organoids and reading out then also changes on the neuronal and potentially also glial response towards COVID. The aims are complemented then by the idea to develop AI-based predictive models to assess the risk of developing NeuroCOV, and as the last step, to identify also drug candidates and laying the foundation for NeuroCOV personalized therapies. Now, the work is in principle is subgrouped into different work packages, and the work package I'm going to talk about today is the work that we're performing with the samples from the two clinical studies, the one in Germany and the one in Italy, where we're performing genotyping of them, collection of blood, and then performing single-cell RNA-seq at the time of recruitment, but also at the time point of 12 months after recruitment to really see what also changes in the individual trajectories of the individuals. And at the same time, colleagues in Milano are reprogramming some of the PBMCs towards iPSCs and are generating organoids and stimulating them. So that we at the end hope to really be able to stratify patients and be able also to understand on a molecular level what effects COVID infection has on the brain and how the cognitive symptoms that Omid just introduced you to are caused, and to potentially then also identify potential therapeutic options. So for the data I'm going to present to you today, so this is still running these trials, so we currently have not yet assessed the full group of individuals, but I'm just going to show to you the first 92 donors that we have now analyzed, and these have been recruited both in Germany and Italy. And as you can see here from the overview of the metadata of the patients, so this is not yet well distributed, so we do have some sites which have more or less equally recruited healthy controls and patients, some others have not yet, but over the time that we've now recruited, so we're now at approximately 400 of patients that have been recruited, this is certainly more balanced as this first batch of samples that we processed. The processing that we're doing in the lab, so this is done by 3 very talented PhD students that you've also seen already in the slides from Omid. They're in principle processing these samples in small subsets of samples, are then running single-cell omics, looking both at transcriptome, surface antibodies of cells, the T-cell receptor and the B-cell receptors. We're also performing flow cytometry and are complementing this with plasma proteomics using 2 complementary technologies, so the Alamar technology as well as Luminex. And what you already can see from this very initial data analysis from this first batch of samples, that we can see trends in the frequencies of cell types in NeuroCOV patients. So we see clearly, or at least trends towards lower numbers of frequencies of monocytes, more CD4 T cells, and also less NK cells, clearly showing that over time and over the number of patients we're recruiting, we will be able to validate these findings, and then could also, based on this already, give some thoughts on what changes really in the individuals having now NeuroCovid. For the single cell sampling and processing, so we were quite happy to see that we could recover now from the first 92 samples approximately 500,000 cells. We're also super happy to see that even that we're recruiting from 4 different sites, we in principle can nicely integrate the data so that we have in the end, and can expect to have at the end, a data set of approximately 600 individuals really that we can analyze together, and can annotate the cell types nicely using the antibodies so that we can see all the major blood immune cell types, and can start to now dig into this data and start to analyze and understand if there are really changes in not only cell type composition, but really also in the molecular profile of these cells, and what you can already see here from this very first interim analysis, that we see changes in gene expression within the different immune cell subtypes, so that we see changes in monocytes and T cells. And when we then further look into this data, I'm just going to flip through this a little bit. So you see when we look at the one side which has recruited in principle homogeneously, so that's Bonn at the moment, then you can see here that you see changes in gene expression between healthy donors and NeuroCovid patients. And when we now try to functionally link this, we see that in the monocytes you see changes in gene expression terms associated with the response to virus, type 2 interferon, cytokine production. So clearly immune-associated terms that are changed in patients with post-COVID syndrome with neurological symptoms. And very similarly also for the T-cell compartment, CD4 T-cell compartment that is also changed in frequencies, we also see very similar changes that you see here also, that in principle within the downregulated genes you see terms that are associated with T-cell activation and proliferation. And you can also see that if you look at NK cells, so when you look there, again, focusing on now one of the recruitment sites, we can see functionally terms associated with NK cell functionality, so cytotoxicity, but also then translation regulation, so that we can say very clearly also here, we expect from this data to see at the end a dysfunctional or proportionally altered NK cell phenotype similar to T cells and monocytes. And we're complementing this, and this is just, we didn't yet go into this, but we're complementing this, of course, with the analysis of the T-cell receptor and the B-cell receptors, to then be at the able to say, and that's also an aspect we discussed already this morning, that if there is an enrichment of specific T-cell or B-cell receptor subtypes, receptor clones, we should be able to see this. Particularly important, I would say, would be here to look at also the second time point to really see what are the changes in the clonotypes. And I'm already seeing that I have to hurry up, so that's the second to last slide. For the NULISA also, or for the biomarker plasma level measurements, we started to look into this. This is for the first 96 donors already done, but here again, this data need more, in principle, more analysis and more individuals to really come to a conclusion of what is changed, but we expect nice data from this. So in summary, the Institute is focused particularly on understanding the neurological and cognitive deficits in post-COVID-19 syndrome. We try to identify single-cell signatures of therapy response and patient stratifications in the National Clinical Study Group, and define cellular or molecular mechanisms underlying NeuroCovid disease trajectories in the NeuroCov consortium. And we see already changes in frequencies and gene expression modules. And with that, I'm at the end. I thank, of course, all the people who are doing the work, the colleagues from the Charité and the NKSG, and of course the consortium, the NEUROCOV consortium, and you for your attention. I'm happy to take questions.
Thank you, Marc. Are there questions from the audience in the room?
Hi, Klaus Diebertin from DZNE. Actually, concerning the single-cell omics analysis in NK cells, did you compare this, or do you have a comparison to other viral infections? And in addition to the age, because NK, also some other myeloid-derived suppressor cells, etc., they changed with age.
So for age, we try to, as best as we can, try to harmonize. We still have, in that first data set, some bias towards younger individuals for the controls. For that question of can we link it or can we compare it to other viral diseases, or that's an aspect where Anna Aschenbrenner will talk about later. So we do have data for this. So we're currently, and will at the end also try to bring these data sets together. I think that should be possible. We're currently also, and that's another aspect, we have focused initially on PCS with neurological symptoms. We're currently recruiting additional patients, PCS patients without neurological symptoms, to also be able to address these questions. Are there specific changes in the cell compartments for NeuroCovid, or is this just a general observation we have, and that the distinction between COVID, post-COVID and NeuroCovid we would do only on the neurological assessments?
One short question, then we need to move on.
Yeah, thanks for this nice talk. I was wondering, you also consider CMV infection in these patients, so that you have, because it's so influential for the immune system and T-cell responses and so on?
So we have not done, I don't think we look at seropositivity for CMV. I mean, with the T-cell receptors, we're able to see it because we see the relevant CMV clones, so that's something, but I think we should consider. I agree. Not yet. So now it's my pleasure to hand over to Anna Aschenbrenner from the DZNE, who is co-chairing the session with me, and I'm very much looking forward to your talk. Thanks a lot.
Thanks, Marc. I will finish up this session by highlighting two more studies where we also looked in the circulating immune cells, as we intend to do and are starting to do for the NeuroCov project. These were actually started even earlier, when it was still unclear if you could even, at least for me also coming from studying acute COVID and having a background in immunology and looking at blood transcriptomes, if we could even see something there. As we know, there are so many various symptoms, and the question is the source of the disease actually in the blood or somewhere else in the tissues? But we know that the blood sample, and of course stratifying patients or monitoring disease and treatment outcomes, could really benefit if we see signals in the blood. So the first study I want to mention was conducted together with Carmen Scheibenbogen from the Charité, as well as Leif Erik Sander and our large team from the DZNE Systems Medicine Department that we have already mentioned. Since we mentioned already also today that the post-COVID patients are very heterogeneous, and considering that we didn't know what signal to expect, we tried to design a cohort that was as similar as possible concerning the patient characteristics. The patients we focused on first here all had only mild to moderate COVID-19, so they were never hospitalized and developed symptoms later on. We sampled them 5 to 12 months after the acute infection, and are looking here at 40 post-COVID syndrome patients and 20 recovered individuals. From those, we took their blood samples, isolated PBMCs, and performed single-cell transcriptomics. In this data set that we generated here, showing you around 230,000 cells, we identified all the cell types as expected. We have a large lymphocyte population in orange to red, myeloid compartment in green, and B cells in purple. And the first question is, of course, if you overlay this now with cells coming from the post-COVID patients and those from recovered, if we see in this low dimensionality reduction shifts in the transcriptomes as indicated but not well mixing of these dots. And we could see this in the monocyte compartment as well for the NK cells. So when we zoomed into these cell types a bit further, now for the monocytes, we can do something called subclustering, so identifying within a cell type certain transcriptional states or programs, and overlaying this again with the disease groups, so the red dots coming from the post-COVID patients, the blue ones from the healthy controls or recovered controls. You can see these shifts, and if you then look into these different substates, you can also quantify that. So we clearly found substates that are either associated with the post-COVID patients or the recovered patients. Another way how to look at this transcriptome data would be by just taking all the monocytes in general and making a differentially gene expression call, and also here on that level we see signatures that can identify these patients compared to recovered controls. The other cell type that showed these shifts on the global UMAP were the NK cells. Also here we saw these shifts. We called the different substates as we did for the monocytes, and when looking at the distributions we certainly find substates that are associated with either of the groups. So in summary, really showing that we can use the circulating immune system to identify these patients. The second project I want to highlight is actually on ME/CFS in general, because one of the major questions we of course have is this now specific to a SARS-CoV-2 infection and post-COVID syndrome, or are the potential other triggering infections postulated for ME/CFS giving a different phenotype or similar signatures? So in this cohort, which we looked at in the consortium of IMI, a BMBF-funded project, also together with the Charité again, and these analyses have been performed by my PhD student Francesco. We have a cohort of 46 female donors who all have different initial infection events, such as EBV, flu, severe infection, one with MISC, and again, some of our post-COVID patients as well. So the ones with the other infection have been sampled previous to the pandemic. When we look at this data, we have produced here roughly 125,000 cells. Again, we can see our cell types that we expect. And concerning now the question on the infection type, we actually see that there are no global shifts concerning our groups when we call them to the infection trigger, meaning that it does not drive clear evident endotypes in the immune compartment that we assess in the blood. But when we zoom into different cell types, and I just want to highlight here also the NK cells, we see actually something similar to the previous project, that also here when we look at the different substates, our healthy controls here in blue have different amounts of one of these substates that we also saw elevated in the other study. So first, confirming across different studies that we see similar things. And again, not huge differences between the viral infection triggers. What we did then is, since now the viral infection did not stratify our patients, we asked the transcriptome data itself, so we made a differentially expression call between all of our ME/CFS patients compared to the recovered controls in each cell type, and made now a heat map of the union of these differentially expressed genes. And what we saw then is that actually all of our patients here indicated on the left side, and the healthy controls have this gray bar here, or the blue one down here, fall into two subgroups. Stratified by the transcriptome. And this is true for each cell type. So if we do this grouping, of course, individually for each cell type based on their gene expression calls, there's not much fluctuation. So they fall in the same group in each cell type that we look. And if we now, I'll try to start this again. If we now take this grouping that was given to us by the transcriptome and compare the other clinical metadata, what we saw was that actually these subgroups are kind of separated by severity based on the PEM score and the Bell score. And the one subgroup with the slightly more severe patients had a longer disease duration, and age didn't make a huge difference. So I think these two projects also highlight very nicely that blood transcriptomes can show the immune deviations in long COVID after mild forms of acute COVID. We also see similar effects in ME/CFS patients, especially in the monocyte and NK cell compartment. The potential infection trigger does not seem to drive these transcriptome-based disease endotypes, but we do see substratification of the cohort with differing in average Bell score. So we are really positive also in context of the NKSG that Marc mentioned and the other trials that we can find a basis also bringing these different studies together that help us to understand how we can stratify patients from the blood and use this then in connection to the clinical trials and see if we can see these endotypes also in the trial cohorts and correlate that to therapy response and outcome. With this, I want to thank you. Of course, this is a huge team effort of our whole department in Bonn and together with our collaborators, mainly in Berlin. And a big thank you to all the funders, especially the BMBF or BMFTR now, and the EU. Thank you for your attention.
Yeah, Anna, thanks so much. Are there questions from the audience? Yes.
Yeah, thank you very much. Really an interesting approach. Do you, I mean, at the end, you may come up with specific profiles for specific patient groups that can be used either to predict outcome or course. Are you already there, or are you planning? What are your plans concerning these profiles that you will get when you do this?
Well, right now, we have started with the different study cohorts and the different projects. First of all, we will try to bring it all together and see how much overlap we have, because we do have post-COVID in several projects, of course, with different inclusion criteria. And I think the nice profiles from the first study I showed, where we really looked at mild, those with a mild acute infection, and then that had, I didn't mention that, they were diagnosed by the Canadian Consensus ME/CFS criteria, so a very specific group without having those that were on the ICU and had severe complications. Those are, of course, very different inclusion criteria to the one we have in NeuroCOV now. First of all, trying to bring that together and see how much overlap we have, either coming up with something common or taking these signature then to the data that we will produce from the clinical trials that Marc mentioned and correlate that to therapy outcome. So if there are these endotypes also there that might show different responses. Because for instance, in the prednisolone therapy trial, maybe we find those with higher inflammation or not, and those are the ones who respond, and those who don't, that won't benefit.
In the DZKJ, one of the main research area is immunity and development of the immune system during aging. And we will also have a program that combines genetically defined immunodeficiency, endotyping common autoimmune disorders. And it might be relevant that one sees in multi-omics approach signaling pathways and deficiencies that are then reflected also in other disease entities not caused by a genetic aberration. So it might be interesting at the end, if you have your cohort together and we have our cohort together, to come up with a combination.
Yeah, very nice.
Maybe just also one comment to what we're doing concerning analysis of the second time point. So we just chatted before that we have currently for that NeuroCOV trial only, I think, for 60 patients for both sides, now reached that second time point, and have not done any of the analysis yet. But from a clinical perspective, impression, gut feeling is that there is changes. To what degree? That's something that we have to certainly evaluate there. But that's something, of course, we want to look at.
So, hello everyone.